Good afternoon, everybody. My name is Steve Van Warmer. I'm the president of PH Aware Global Association, and we are very excited to have everyone here with us today to kick off our On Air with PH Aware summer series of webinars that explores the remarkable evolution of pulmonary arterial hypertension from its earliest challenges to today's breakthroughs and the promising future of research and care. Whether you're a patient, a caregiver, a healthcare professional, researcher, or advocate, these live conversations are going to offer valuable insight into how far the PH community has come and where it is headed to next. And I was very lucky enough to be in the standing room only session at the President's Symposium at the kickoff of ATS 2026 in Orlando earlier this summer, which traced the history and plotted the future of PAH therapeutics pathways. And the speaker who kicked all this off is today's presenter, Dr. Sharon Rounds. Dr. Rounds is a nationally recognized pulmonologist, physician scientist, educator with decades of leadership in the pulmonary vascular disease space, critical care, and translational research. She serves as professor of medicine at the Warren Alpert Medical School at Brown University, where she has held senior leadership roles advancing clinical and translational research. Dr. Rounds is a former president of the American Thoracic Society and a fellow of the American Thoracic Society, an honor recognized for her significant contributions to pulmonary medicine and respiratory research. An internationally respected expert in pulmonary vascular disease, Dr. Rounds has helped shape this field through her research, her leadership, and service on national and international committees. She served on the American Thoracic Society Committee and developed the landmark statement on pulmonary hypertension phenotypes and has been instrumental in advancing collaborative research efforts that have improved the understanding, diagnosis, and treatment of pulmonary hypertension. And following Dr. Round's presentation today, we're going to have time for some of your questions. So please feel free to put those in the Q&A tab at the bottom of your screen there or in the chat, and Dr. Rounds will answer those after her lecture. And by the way, we are very happy to have her today because I woke up to an email this morning that she had catastrophic engine failure of her car. I wasn't sure if she was going to make it today, so she's joining us live from a Toyota dealership on the East Coast where she's about to purchase a brand new car. So I cannot thank you enough. But one other thing I want to say, a big thank you to our webinar sponsors today, Merck and Liquidia. Through the strategic use of innovative technology and compelling storytelling, your support helps us here at PH Aware amplify the voices and experience of patients worldwide, fostering a more informed and connected patient community. So without further ado, I welcome you to the early days, the origin of PH therapies and trials. Dr. Rounds, again, thank you for joining us here today.
Okay. Well, thank you for that kind introduction, Steve. It's a real pleasure to be able to kick off this summer series that you've organized. I have to say that I am, feeling my advanced age when I look back over the literally the decades of remarkable change in this field. So what I'd like to do today is to kind of review some of what I consider to have been really landmark events in the early development of pulmonary hypertension therapies and also understanding of the disease. And I would like to be able to share some of the comments that Steve was alluding to from the presentation that I had the pleasure of making last May. So bear with me for a few slides, if you could, please. And what I was tasked with by Riad Dweek, who's the current president of the American Thoracic Society, to talk about the origins of pulmonary hypertension therapies. And when I thought about that, I realized that it was more than just development of treatments, but it was also development of understanding of a disease. And so that's what I'd like to kind of outline today. So, at the very beginning, I have to say I have no disclosures, no ties to pharma, but I do have to apologize in advance because of course, we can't acknowledge everyone who has made so many wonderful contributions to this field. I'm very grateful to my friends and colleagues, Norbert Vokel, Greg Elliott, and Gazwan Boutros, who have recently put together a series of conversations and books with their recollections, and that was very useful knowledge for me as I was thinking about this. And I also want to make a note of the wonderful leaders in this field who were so important early on, whom we've lost. One is Robin Barst, who is a pediatrician at Columbia University, who really put pediatric pulmonary hypertension on the map and made sure that the world understood that this was a disease that also affected the very young. Jane Morse and John Newman were the discoverers of originally of the gene for familial pulmonary arterial hypertension. And Guy Zimmerman from the University of Utah was a leader in our understanding of the basic biology of the cells involved in the pulmonary circulation. Very sad that we have lost them in the last few years. So what we'll be doing today is doing a history lesson. And these are the things that I'd like to review. So pulmonary hypertension, which was originally called primary pulmonary hypertension, was first described in a case report in the German literature in the late 1800s, excuse me. But the first comprehensive description of the clinical aspects and the physiology was published in 1951 with a group of patients from New York City. This was followed in 1970 by a very detailed definite description of the pathologic changes in the lungs of these patients with so-called primary pulmonary hypertension. And two really important points came from this pathology description. One was that this disease involves the entire pulmonary artery, and that including both the intima, the media, the vascular smooth muscle, and also the adventitia, the fibrous tissue surrounding the vessel. So all three layers of the vascular wall. Similarly, this paper described very characteristic pathologic findings of these very abnormal cells, endothelial cells, that normally line the pulmonary arteries, were clumped together in this lesion they called plexiform lesions. And this is a very characteristic and diagnostic pathologic change. So this was a very, very important breakthrough. Not only do we now have an idea of the clinical aspects, the physiology, but we also have an understanding of the changes in the cells within the lung vasculature. Sorry, I need to advance here. Why can't I do this? Here we go. So in 1960, the World Health Organization organized a meeting of international experts on what was called chronic cor pulmonale, or chronic heart failure due to lung disease. And this was chaired by a Nobel Prize winner, Dr. Dickinson Richards, who won the Nobel Prize for doing one of the first right heart catheterizations at Columbia. And this World Health Organization defined cor pulmonale and classified it due to diseases of the lung or of the pulmonary vasculature. They had also a call to action for training of investigators and a call for additional meetings to share and disseminate information about this disease. And this last point is very, very important because international collaboration has been really critical in developing our understanding. In the 1960s, unfortunately, what was then considered a rare disease became an epidemic in Europe. And this was due to use of an appetite suppressant, Aminorex, which is a pharmacological analog of amphetamines. And people died. And this Aminorex epidemic was the stimulus to the second World Health Organization meeting on pulmonary hypertension, which was held in Geneva in 1973. And at this meeting, they called for a study of the natural history and pathology and family studies of pulmonary hypertension. They advocated for simple, non-invasive screening methods for early cases. They called for a central registry of cases and also made note of the importance of what they called pharmacovigilance. In other words, being careful when drugs are introduced to the market that they do not have bad side effects on the lungs. However, despite this, in the 1990s, there was yet another epidemic of pulmonary hypertension due to appetite suppressants, this one due to so-called fen-phen, which was a cause of considerable morbidity, even mortality. So the fen-phen epidemic led to the third World Symposium on primary pulmonary hypertension in Evian, France in 1998. Stuart Rich, who is a phenomenal leader in our field, led this World Health Symposium, and it was at this symposium that the currently used system for nomenclature and classification of pulmonary hypertension was developed, and it's subsequently been improved upon, but basically this originated in the late 1990s. So the take-home message from all of this is that international meetings set the standards for clinical research. And I can't emphasize the importance of the international collaboration. This includes now continued World Health Organization meetings, the Pulmonary Circulation Assembly of the American Thoracic Society is very important in these as an international scientific meeting. There are Grover conferences in the pulmonary circulation, and of course the Pulmonary Vascular Research Institute now has international scientific conferences and educational events. So I mentioned that one of the World Health Organization symposia called for a national registry. So in 1981, Dr. Al Fishman from University of Pennsylvania and Claude L'Enfant, who at the time was director of the Division of Lung Diseases at the NIH, organized a U.S. national registry of cases of primary pulmonary hypertension. This registry was led again by Stuart Rich, who was the principal investigator on the grant that funded this. There were 32 centers across the United States. Some of the site PIs went on to continue to work in this field. We were all very young then, just getting started, And I want to tell you, it was so exciting to work with such a talented and interesting group of enthusiastic physician scientists. The registry prospectively enrolled 194 patients. There were very, very strict diagnostic criteria. The goal here was to understand what we call the natural history of this disease, the pathology, and to explore the effects of various treatments. The patients were followed for 7 years until 1988. And let's see here. We need to advance. And the results were originally reported in 1987. And what we see here from these results is that this is a disease that spans the age of patients from the very, very young to much older. It affects both males and females. At that time, the mean pulmonary artery pressure of the participants was quite high, 60 millimeters of mercury. And unfortunately, it was a very short duration of time from symptom onset to first diagnosis. But it could take as long as 2 to 4 years for patients to finally get a diagnosis. Survival in this registry at that time, when there were no effective treatments available, was about 50% in 4 to 5 years. Really pretty dismal survival, which was quite discouraging. So the take-home message here is that these case registries were the beginnings of the development of multi-center trials, and collaboration has been absolutely critical in our understanding of both the disease and its treatments. Now, of course, there are multiple registries of patients, including both in this country as well as internationally. And they're all very important in helping us understand what's going on. I'd like to kind of switch gears a little bit and point out that at the same time that we were beginning to understand the disease, there was also a growing understanding of the physiology of the lung blood vessels. And a place that I think was very important in the development of this understanding was the Cardiovascular Pulmonary Laboratory at the University of Colorado, which was led by 3 really important scientists, physician scientists, Jack Reeves, Bob Grover, and Ivan McMurtry. I, many years ago, had the pleasure of training there at the same time as my friend John Newman and also Norbert Vokel. We, at that time, were doing studies on rodents, rats, who were exposed to hypoxia to cause pulmonary hypertension. The CVP lab was a terrific place to work. We worked hard, but we had a great time learning about the disease. It was an incubator of great ideas, and every day the laboratory leaders would walk through the lab and ask each of the fellows, what did you learn today? And it was a great day when you could tell, oh wow, today I learned X or Y. And this was a really exciting time. One important observation that came from the CVP lab was the fact that calcium channel blockers drugs that block the entry of calcium into smooth muscle in pulmonary arteries were effective in blunting pulmonary pressure responses. And here we see pulmonary pressure response in millimeters of mercury versus time with an isolated blood-perfused rat lung. And when the animals, when the lungs were ventilated with hypoxic gas, pulmonary artery pressure went up, which is a expected response. But this response was absolutely knocked out cold by verapamil, a calcium channel antagonist. And so this very important animal observation was picked up by Stuart Rich and colleagues. And what they found subsequently was that, in fact, there were some patients who were very sensitive and responding to the effects of calcium channel blockers. And here they report the effects of high doses on survival in primary pulmonary hypertension. And the patients who were so-called responders had very good survival with treatment with calcium channel blockers, whereas non-responders had the same unfortunate prognosis as those from the NIH registry. So we now understand that this calcium channel blocker responsive phenotype accounts for 5 to 10% of patients with pulmonary arterial hypertension. And they're identified by vasodilator challenges during right heart catheterization. There's likely a genetic basis for this, and this was really the very beginning of a so-called personalized medicine approach to treatment of pulmonary hypertension. But unfortunately, it's only a very small percentage of patients who are responsive. So the take-home message here is that findings in animal models translate to successful therapeutics in people. So it's very, very important to continue the basic research with cells and small animals in addition to clinical trials in humans. Another important observation that came out of the University of Colorado was the observation that some people and some animals are very sensitive to high altitude. And this cow actually has pulmonary hypertension. It's so-called brisket disease. And the cow has heart failure from pulmonary hypertension due to residence at high altitude in Colorado. And here the peripheral edema has collected in the front of the cow, the so-called brisket area. And in fact, Bob Grover and Jack Reeves actually brought both cows and lambs to the top of Mount Evans in Colorado, which is 12,700 feet. And after a summer at 12,700 feet, the cows developed very high pulmonary arterial pressures, whereas the lambs did not. Thus, there seems to be some genetic susceptibility to the effects of high altitude on the pulmonary circulation. So this animal observation has formed the basis for subsequent work where actually the gene for high altitude pulmonary hypertension was described, and John Newman from Vanderbilt was very important in leading that effort. And John and Jim Lloyd, also from Vanderbilt, described heritable pulmonary arterial hypertension occurring in numerous families. And one really interesting aspect of this is that what they found is if they went back 1, 2, 3, 4, even 5 generations, individuals who were thought not to be related at all. In fact, if we went back far enough, actually had common founders. And so what was then thought to be sporadic pulmonary hypertension actually frequently actually turns out to be inherited or familial. So John and Jim, as well as Jane Morse from Columbia University, in 2000, both almost simultaneously reported the gene for familial pulmonary arterial hypertension, the bone morphogenetic protein receptor type 2 gene. And here we see the groups of discoverers all celebrating with champagne in Venice at a WHO symposium in 2003. So the point here is that the Human Genome Project, which was sponsored by the National Institute of Health, plus family registries and DNA archiving, were very important in identifying genes for pulmonary arterial hypertension. And now, of course, we know that it's not just BMPR2, but there are a number of genes that are important in this particular disease. So the take-home message here, again, interdisciplinary international collaborations resulted in remarkable progress, and that the animal studies formed the basis for important insights into the disease in humans. Now, switching gears slightly, my own personal scientific interests have been in a particular type of cell in the lung called the pulmonary endothelial cell. These cells line pulmonary blood vessels, and I was very grateful to my former boss, Jerry Brody, for encouraging me in pursuing this interest, which is, I think if I contributed anything to the field, it was to better understanding of how endothelial cell injury might contribute to this disease. The lung endothelium, of course, is a metabolically active tissue, and many investigators have contributed to that, including Una Ryan, who was the first person to isolate endothelial cells from the lung and maintain them in tissue culture so that they could be, their biology could be studied in fine detail. We now know that lung endothelial cells are important in all sorts of functions of the lung, including regulating permeability to water, inflammation, detoxifying drugs, metabolism, CO2 excretion, and many, many others. And these are just a few of the people who have contributed to this field over the years. So, again, a take-home message that different experimental models and designs have all been important in furthering our understanding of the disease, ranging from cultured cells to animal models to human observational and epidemiology studies and to clinical trials. They all add unique knowledge and insights. So, you know, I hope I've made the case that interdisciplinary international collaboration is critical to defining and understanding pulmonary hypertension, that basic discoveries translate to discoveries in people, and that it's really important when we look toward the future to not underestimate the power of a new idea. And in this case, the new idea was that there were diseases that uniquely affected the lung blood vessels, and we needed to understand them more. So I'm going to stop there and try to stop sharing up. Perfect.
That was great. No, oh, sorry.
Did the job. So anyway, Thank you, everybody, and I'm happy to entertain any questions or comments.
Yeah, thank you so much, Dr. Rounds. Awesome. So yeah, anybody that's listening, viewing here, if you see in the bottom of your screen, there's a Q&A tab. If you have any questions, feel free to drop them in there. And but in the meantime, I prepped a few questions for you, Doctor, if you don't mind.
No,
But you know, what I find so interesting is that, many of these earliest studies, you said dating back to the 50s, 60s, and your time as well, involved incredibly small patient populations, limited resources. Did you have any sense that in the Wayback Machine there, that foundational studies that you were doing and you and your colleagues would eventually transform the field as great as it has to today?
well, neither I nor anyone else has a crystal ball, of course. I think, there's nothing like success to stimulate success. And once we had one drug that worked, then all of a sudden there was great interest in finding additional drugs. But before there was anything, it was very discouraging because you kind of hated to make the diagnosis because there wasn't much to offer. So success stimulates success. And I think it's just astonishing how the field has blossomed in the last 30, 40 years. It's remarkable.
And kind of springboarding on that, today's clinicians have, you know, dozens of clinical trials, multiple drug classes, sophisticated diagnostics, risk assessment tools, all these kind of things. How extraordinary is it to witness just
Oh,
that
it's
from your seat,
incredible.
the transformation?
It's incredible. And I don't want to underestimate the importance of the influence of patients and caregivers here. Because, one thing I learned when I was in the leadership of the American Thoracic Society is the critical importance of dedicated patients and stakeholders in both in stimulating and supporting and inspiring scientific advance. The American Thoracic Society has something called the Public Advisory Roundtable, with representatives from patient interest organizations for a number of diseases, including pulmonary hypertension. And the, 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 the patients have been an inspiration, and also, they've been challenging. We want the cure, and they mean it. And God love you. Good work.
Yeah, it is interesting, in my family, in all our families, we've had people that are impacted by different um, disorders and diseases. But what do you think that is so unique about this close collaboration that I've witnessed in all these decades here in the pulmonary hypertension space?
Well,
Unlike something else.
well, I think pulmonary hypertension has, been particularly successful in engaging patients and families. Um, the the thing I think that has been really wonderful is that people are understand the importance of clinical trials. They're willing to sign up for clinical trials. They're willing to participate, and without that collaboration and cooperation, I mean, the field would be set back decades. So thank you for all that you do to help bring treatments to the public. It's wonderful.
And there are clinicians, just entering this field today that we'll never know a world that didn't have pulmonary hypertension treatments and therapies. Like, what kind of lessons from your early years and your colleagues that you outlined here do you hope that this new generation and the generations that follow don't ever forget?
Well, I think one thing that I think we've tried very hard to do all along is provide good supportive care. I mean, it's one thing to have a drug, a pill, or infusion. It's another to have group, a clinician who is looking at the whole patient and providing support for maintaining physical activity, for example, good nutrition, patient education. I think that, you know, I certainly hope, and I'm sure that clinicians will continue to provide what we call good supportive care in addition to, prescribing medications. It's all part of the picture and an important piece of the picture. And back in the olden days, we didn't have anything but good supportive care. That's all we had.
Yeah. You mentioned earlier you and Robin Barr's work in the field early on as one of the few females in the field. You named a couple others along the way, but did you have just any personal barriers or challenges
Oh
as
boy,
you
yeah.
evolved, or was that pretty? I don't know. What was that? What was that like at that time?
Well, I tried not to let it bother me. But yeah, it was a little lonely for a few years. But one thing that has been really terrific uh, is that I do think that the pulmonary hypertension provider community is an inclusive one, and the people are welcomed and appreciated for their contributions, regardless of their sex or national origin or whatever. It's a very inclusive community.
Yeah. Well, let me ask you just a couple more. Like when you really reflect on this whole evolutionary period of PAH work and therapeutics, from starting with a disease that had a little more than supportive care, as you mentioned, to having multiple life-extending therapies, which one of the benchmarks in these at-home takeaways, as you mentioned, which one of the are the ones that is the one that you're most proud of?
You mean in the field as a whole?
Just in the field, or like the big solution or thing that happened that was okay, this was the game changer that really set things off to the races.
To be honest, I think it was enhanced understanding of the function of the cells in that comprise the pulmonary blood vessel. And as we understood the function of endothelial cells, vascular smooth muscle, etc., then drugs could be designed in a rational manner. So without that basic understanding, I think we would be decades behind.
Yeah. Well, to that end, if as someone who's really witnessed the whole modern evolution of research in pH, what gives you your greatest optimism now, decades later, that and you're kind of as you look into you know the the future here, the decade ahead, what challenges do you think really needs to be solved?
Well, I think precision medicine is where we're headed. We not, there's a lot of individual variability, not only in genetics, but also in physiologic responses. And I think, we have a long way to go to catch up with some of the advances that have been made in understanding precision medicine and cancer. My hope is that we'll be able to use the same insights and tools to be able to design individual therapeutic regimens that minimize side effects and maximize gains for each individual patient. I think that's where the future lies, in my humble opinion.
Wonderful. Well, to anyone else, I don't see any other questions unless you have any others, folks. I'll kind of let Dr. Rounds get to closing that car deal. But if I can share my screen for one moment, though, however, I just want to mention to everyone listening that we're going to continue this webinar series all through August. The next one is 2 weeks from now with Dr. Val McLaughlin, who's going to really talk about the birth of modern PAH therapies. Then combination therapy, redefining PAH treatment strategies with Dr. Sandeep Sahai on August 17th. And really kind of looking towards the future of emerging targets and therapies with Dr. Stephen Chan on August 19th. And finally, this August, we'll close out with Global Centers of Excellence, Collaboration and Care with Dr. Benicio de Jesus Perez from Stanford. And I do want to say, with regards to this webinar, once we're concluded here, we'll put this out and make this available on our socials and on our HeartWorks app. And I'm going to leave this up for 1 second. If you're not familiar with our HeartWorks app, this is a patient. It's a research and resources app that features not only our podcast series, 600 some episodes, a patient lived experience portal with the chatbot features, clinical trial matching tools, CME education for all these for doctors and nurses that might be joining us here today, and remote 6-minute walk tests, etc. News feeds from all over the globe. So I'll leave this up for a moment. You can scan the QR code to download it in the Apple Store or at Google Play. But beyond that, let me see if I can. I'm going to minimize this now so I can get back to my screen. And I'll stop sharing and say, again, thank you so much, Dr. Rounds, for being here today, and everyone for attending. We appreciate you so much. And once again, I thank you to our webinar sponsors, Liquidia and Merck. So we'll see you next time, folks. Thank you so much.
Bye-bye.